everyone. I am Dr. Chatterjee, consultant ENT and head neck surgeon. And in this video, I will discuss about the development of the external ear. So to start with the development of the external ear, we need to understand what are the parts of external ear. So there are namely three parts of external ear. Starting off with the auricle or the pinna. Next, it is continuing as external auditory canal ending at the tympanic membrane. Clear? So in this slide, I will just mark the parts of the auricle or pinna, and in my upcoming video I will discuss in detail about the anatomy of the external ear. So here we will only identify and label the parts that is required for studying the development of external external ear. Okay, so let's start. So to start with, this structure, this outermost cartilaginous structure is known as helix. This cartilaginous structure is continuing here. The cartilaginous structure that is just medial to the helix is known as antihelix. The cartilaginous structure which is the most medial portion, this is the tragus, and this structure is also cartilaginous. This is known as antitragus. There is a projection in the helix. This projection is known as Darwin's tubercle. This is known as Darwin's tubercle, and it is an atavistic part. Atavistic part, that is at the part of development from our previous generations as well as during the evolution process, but it has not got any clinical significance so far. Now I am highlighting a part. This is the antihelix. Now this antihelix is going upwards and bifurcating into two structures. These two structures are known as the crura of antihelix. Clear? In between these two crura, there is this yellow triangular region. This yellow triangular region that I have marked is the fossa triangularis. This is the fossa triangularis. Now the helix is continuing downwards and ending at this point, and this is known as the cross of helix. So this is the cross of helix. Okay, now between this cross of helix and the beginning of the tragus, and cross of helix, this point which is free from any cartilage is known as incisura terminalis. Incisura terminalis. Okay, and there is another cartilage free area here between the antitragus and tragus. This is known as incisura intertragica. Clear? And this portion below is the ear lobule, which is filled with fat. This is the ear lobule. The portion between this inferior crust of antihelix and the crust of helix, this marked portion is the Simba concha, and this structure, this region, this region that is continuing into the external auditory canal is the cavum concha. It is the cavum concha. So this is in brief the parts of the pinna or the auricle that is required for the study of development of the external ear. Now here this is the here we will understand the in brief the anatomy of the external auditory canal. So external auditory canal starts at this point and ends at the tympanic membrane. So this is the portion of the external auditory canal, and this is in total 24 millimeter long. This is in total 24 millimeter. That is EAC, external auditory canal. It is divided into two portions, the lateral 8 millimeter part and the medial or inner 16 millimeter part. Clear? So this lateral part is 8 millimeter and this medial portion is 16 millimeter. Clear? So this 8 millimeter part is cartilaginous portion, and this inner 16 millimeter is bony portion. So lateral one third, that is 8 millimeter portion is cartilaginous and inner 16 millimeter is bony portion, and it is ending at the tympanic membrane. So another important point, it's an MCQ point. Another important point, it's an MCQ point, is external auditory canal is the only cul-de-sac in our body, only cul-de-sac in our body that is lined by skin. Clear? So it is the only cul-de-sac in our body that is lined by skin. This is an MCQ point. Now in the next slide I will discuss about some of the important points which is important for the study of the development of tympanic membrane. So we will study some of the important highlights of tympanic membrane. So point number 8, tympanic membrane separates external ear from middle ear. Very basic point. Second, has two parts. One is pars tensa, another is pars flaccida. We will discuss in detail the anatomy of each and individual parts of external ear in my upcoming video. And tympanic membrane has three layers. The outer layer is the epithelial layer. The middle one is the fibrous layer, and the inner layer is known as the mucosal layer. Clear? So this is very important. These 3 points, it separates the external layer from middle layer, right? Two parts, pars densa, pars flaccida, and has 3 layers. So parts and layers are different, mind it. Epithelial layer, fibrous layer, and mucosal layer. Clear? Now, to start with the development of the external layer, the ear develops in the region of lower neck initially, in the region of lower neck. The mesenchymal proliferation takes place upwards in a posterolateral direction. Posterolateral direction. So, if this is the neck, the ear develops in the lower neck and goes posterolaterally to attain the final position. Okay, to attain the final position. Clear? Now, these during the 4th week of intrauterine life, during the 4th week of intrauterine life, these 6 tubercles develop from the mesenchymal proliferation of the first and second pharyngeal arch or branchial arch. Okay, so these 6 tubercles develop from the mesenchymal proliferation of the first and second branchial arch, and these tubercles are known as tubercles of His, or they are also known as hillocks of His. They are numbered as 1, 2, 3, 4, 5, 6. Okay, they are numbered as 1, 2, 3, 4, 5, 6. It is stated that these three, the first 3 tubercles develop from the first branchial arch, and the second 3 tubercles develop from the second branchial arch. But there is another controversy regarding this point. So this is according to new theory. This is according to new theory. Okay, this is the new theory. If we go in accordance with the old theory states that this first tubercle only develops from first branchial arch, and rest of these 5 tubercles develop from second branchial arch. Clear? Now, as according to recent journals, this theory is proposed everywhere, we will take into consideration this theory. But if we go by concept, as the auricular muscles are supplied by facial nerve, which is a nerve of second branchial arch, this theory seems to be more conceptual. But all the recent journals are stating that this theory is worldwide more accepted. Okay. Now, From the first arch. First arch develops tubercle 1, 2 and 3, and they form the structures tragus, helix and Simba concha. And the second arch gives rise to tubercles 4, 5, 6 and they form cavum concha, antitragus and antihelix. Okay, so if I go back to this slide, and erase these portions, I will denote the portions that is developing from the first arch and the second arch from the in accordance with the new concept. Now, so from the first arch developing is what is developing? Tragus. So this portion, helix, this entire portion and Simba concha, this portion. And from the second arch is developing antihelix, which is this portion, antitragus, which is this portion, and cavum concha, this portion. So this is from the second arch, and this is from the first arch. Clear? And this is from the first arch. Now it is very important. So these portions form these structures and they fuse to give rise to the adult ear. So the adult ear develops by the third month of IUL, intrauterine life. Third month of intrauterine life. Clear? By the third month of intrauterine. Now next we will discuss regarding the development of external auditory canal. So here we will study the development of external auditory canal. Now external auditory canal starts developing as a prolongation of the first ectodermal cleft. First ectodermal cleft. This prolongation takes place between the first arch and second arch. first arch and second arch. In between that, the prolongation occurs. Now, the prolongation occurs towards which structure? The prolongation occurs towards the structure which is known as the tubotympanic recess, TTR. This TTR is tubotympanic recess, which is giving rise to middle ear structures and pharyngotympanic tube, that is eustachian tube, tubotympanic recess. Now, as this structure develops, this first ectodermal cleft towards the developing middle ear structures, there is an epithelial plaque formation in the medial portion. This is an epithelial plaque formation. Okay, and this is known as the meatal plaque. This is known as the meatal plaque. Okay, this is known as the meatal plaque. Now, this plaque gets canalized. This plaque gets canalized between the 8 to 10 week of 8 to 10th week of intrauterine life. This plaque gets canalized. Plaque gets canalized. Okay, to form the external auditory canal. Now, between this portion, that is the developing tubotympanic recess, that is forming the middle ear structures and pharyngotympanic tube, and this external auditory canal, there lies a mesenchymal layer. So this is in particular is a mesenchymal layer. So this is in particular is a mesenchymal layer. So we can see that it involves contributions from this ectodermal layer mainly, which is ending at this mesenchymal, touching the mesenchymal zone. And this meatal plaque gets canalized between the 8 to 10 week of intrauterine life. So this is the portion, that is the MCQs also come from this portion, and from the university examination point of view also this much is enough for development of the pinna or the auricle and development of the external auditory canal. Now we will go towards the development of the tympanic membrane. So we will study about the development of the tympanic membrane. So tympanic membrane, so as we have seen already, that is the ectodermal cleft was coming first, and in this part there was the developing pharyngotympanic tube, and in between that there was a mesodermal layer. There was a mesodermal layer. So this is the, so this overall, now if we already only zoom this area, we will be able to understand that tympanic membrane has three layers. As we have already studied, outer layer is the epithelial layer. Epithelial layer. Middle one is the fibrous layer. And the inner one is the mucosal layer. So we have already studied this. Now this ectodermal lining forms this epithelial layer. So it is coming from epithelial layer, E for E, ectodermal. Ectoderm. Okay. This fibrous layer is coming from the mesenchyme, which is sandwiched between these two structures. That is tubotympanic recess, ectodermal cleft. Ectodermal cleft tubotympanic recess. Okay, so this is the fibrous layer which is developing from mesoderm, and inner mucosal layer, it is from this endoderm. So important point is that tympanic membrane develops from all these three structures: A, ectoderm; B, mesoderm; C, endoderm; D, all of the above. So the MCQ answer will be this one, all of the above. Okay, the answer will be this one, that is all of the above. Hmm. So here, if we give a small recap, what was the objective of this session? The objective of this session was to study the development of external ear. We have already seen the parts that I have just erased to demonstrate this slide. Parts of auricle I have described in the previous slide. Here we can see this is the external auditory canal, the parts being cartilaginous and bony, and it is the only cul-de-sac that is in our body that is lined by skin. Next, the tympanic membrane, two parts, flaccida and tensa, three layers: outer epithelial layer, fibrous layer, mucosal layer. Here we have studied about the hillocks of His or the tubercles of His. The ear develops generally in the lower cervical region, and that extends upwards posterolaterally to form to attain the final position, right? To attain the final position here, the tubercles of the His and the hillocks of the His, the 6 in number. First three develops from first branchial arch, and second three from second branchial arch, right, according to the new theory, obviously. And if we go back to our slide, so what are the structures that is arising from the first arch and second arch? The portion labeled with the sky blue color develops from first arch, and that with the pink color develops from second arch. So the sky blue color demonstrates the helix, the tragus, and say Simba concha and the cavum concha, antitragus and antihelix being developing from the second arch, right? Next, we have come to the development of the external auditory canal, obviously, which shows that it develops as a prolongation from the first ectodermal cleft that gets plugged medially to form the epithelial plug or meatal plug, and that gets canalized between the 8th to 10th week of intrauterine life. And the developing tubotympanic recess forms the middle ear structures and pharyngotympanic tube, and in between this ectodermal cleft and tubotympanic recess lies the mesodermal layer. Now in next slide we have discussed about the development of tympanic membrane. It has three layers: epithelial, fibrous and inner mucosal layer. Epithelial layer develops from the ectodermal cells, fibrous layer from the mesodermal cells, and obviously endodermal cells or endodermal layer giving rise to the inner mucosal layer. So the tympanic membrane develops from all these, has contributions of all three: ectoderm, mesoderm and endoderm. So we have come to the end of our discussion, which was only restricted to the development of external ear. In my next video, I will talk about the anatomy of the external ear, because these portions form the basis of the study of ENT, means ear otology. So the portion of ENT that deals with the study of ear is known as otology, and so that forms the basis of otology. Just understand the development and study the anatomy, because the further diseases will need concepts of these things. So I have started with this. Thank you for your patience hearing. Thank you. Good night.